Hello, everyone, and welcome to Manomet Small Seed webinar series. My name is Andrea Ferreira, and I'm Manomet Science Communications Manager, and I'll be your host today. In this edition, that I'm very excited, I can't be biased, we have Clare Cunningham, Senior Education Manager at Manomet for our program on environmental education and outreach. And we're gonna talk about, she's gonna talk, she's the expert, not me, about Songbird Science and Stumpers. A few guidelines now we begin. As you can see in the screen, there are a lot of buttons that you can see on the part on your screen, or you can have captions if you want to in different languages. Also the chat, and if you have any questions, you can put them on the Q&A box right there so we can answer them at the end of the small set. Also, this session is being recorded for you to know. Even if your friends are not joining in, you can send them the link later. It will be on our YouTube channel. We would like to know where everyone is joining us from today in this Zoom. So if you can just go in the chat and type in where you're at, say hi to Clare, and we will welcome this session because I want to dedicate all the time to Clare and all of the exciting things that she has to say. Welcome, Clare. The stage,
Hi.
the virtual stage is yours.
All right. Are we ready?
Yes.
Awesome. Okay. So, hi, everybody. I'm Clare Cunningham. I'm the Senior Education Manager here at Manomet Conservation Sciences. And a large part of my work involves the coordination of school group visits from all over southeastern Massachusetts to visit the Trevor Lloyd-Evans Banding Lab, which is where students come to take on the role of banders while also analyzing and interpreting Manomet's own banding data. And just to be very clear, students do not handle the birds or band the birds, but we do use models to simulate similar experiences. And each year we see more than 1,200 students come through the banding lab to learn about our work. And so we thought that today we would share some of the magic of the banding lab with you all and also sprinkle in some trivia questions. So if you would like to participate in the trivia portion of this, please grab some paper and a writing utensil, and you can keep track of your score as we go, and then we can share them at the end. And we are using the honor system, so please be honest. So Manomet's Banding Lab is on the northeast coast of Massachusetts, and it overlooks Cape Cod Bay. So if you think of Cape Cod as an arm, you're looking right into the elbow pit of the bay. For several reasons, this is a really ideal location for a banding station. And I'm wondering if you all know which coast or which major flyway the banding lab sits on. And I'm going to give you 10 seconds to write down your answers. Okay, 10 seconds is quick. So we are sitting on the Atlantic Flyway. There are three major flyways that span across the hemisphere, and they can be broken down into smaller sections, but the major flyways are the Pacific Flyway, which follows the Pacific coast, the Mid-Continent Flyway, which goes right down the center of the hemisphere across the Americas, and then the Atlantic Flyway, which follows the Atlantic coast. So these flyways are essentially like super highways for birds that are connecting them from their breeding grounds to their non-breeding grounds, and they have really critical stopover sites with ample food and appropriate habitat along the way. Um, so here we are on the East Coast in the Atlantic Flyway, and the Trevor Lloyd-Evans Banding Lab is one of the longest continuously running migratory banding stations in the country. So we've been collecting data the same way since its beginnings, and our site location and data collection methods have stayed relatively unchanged since we started. And I'm wondering if anybody here knows, when did we start banding birds at Manomet consistently? Was it A, in 1965, B, 1959, C, 1969, or D, 1975? And I'm going to give you 10 seconds. All right, 10 seconds is up. So if you guessed 1969, you are correct. And that means that we have just under 60 years of banding data showing how the timing of migration and population numbers have changed locally over time, which is really, really incredible. Um, and before we can even start thinking about banding our birds, we need to go out and catch them. So I'm wondering if you all know how we go out and catch birds. Is it A, do we play songs that lure the birds into big giant nets? B, do we set up nets hidden by brush that birds fly into as they move around throughout the day? C, do we go out on trails and catch them with essentially butterfly nets while they're resting? Or D, do we put out bird food and throw sheets over the birds when a flock comes in? You have 10 seconds. All right. So I know that some of these sound really strange, but a lot of people do use songs to lure in birds, and this is called playback, and it often is used for targeting specific bird species. So we will do this sometimes when we're banding saw-whet owls. We'll play saw-whet songs and lure them into the mist nets. But for the most part, Manomet is a passive banding station, which means that we don't use any callback to lure birds into our nets, and we're really interested in just learning about the birds that are flying through the property on their own. So we do use large nets that are hidden in brush, and the birds will fly into them naturally as they move around throughout the day. And we have two major banding seasons, so one in the spring and one in the fall that line up with migration, running from April 15th to June 15th and August 15th to November 15th. Answers C and D here were both suggested by students, and I have to say that C sounds absolutely exhausting, and D is not very ethical. So we don't do either of those things. Um, but the mist nets that we use at Manomet are about 10 feet tall and 40 feet long. So on the right, you can see some of our banders standing next to them, and the nets become essentially invisible when they're fully extended. So birds will fly into them without seeing them. And we have 50 net lanes along the property or 50 nets along the property, and it ends up being about a mile of what we call net lanes. Um, and so on the left-hand side, we've got one of our banders from a couple years ago setting up a mist net, and then we have our throwback photo of somebody setting up a mist net using almost identical form. So it just goes to show you how our methods have stayed the same over the years. Um, so once the nets are set up, our banders go out and check them every 40 minutes to see who has flown into them. And the mist nets are made of a really, really fine material that just gently tangles the birds without hurting them. And I like to think of it as like a giant hairnet that they fly into. And there are different size nets for different size birds. So for smaller songbirds like ours, we use a really fine net. But if you're targeting larger birds or shorebirds, you might have a slightly thicker net to capture those birds. And again, on the left, we've got a black-cap chickadee that's being extracted from a mist net, and that's one of our archived photos. And then on the right-hand side, we've got a white-throated sparrow being extracted from our bog nets, and of course, an American goldfinch. So I have a question for you all. If you have access to a mist net, you have the supplies, and you have the space, can you go out and catch birds? True or false? You have 10 seconds. Or yes or no. All right. So it is false. You cannot just go out and catch birds if you want to. All of the bird banders that are at Manomet and all across the hemisphere are federally permitted and have permission to be banding birds and extracting them from mist nets. So you can't just decide one day, oh, I've got the equipment, I want to go catch some birds. Typically, Manomet will have two lead banders on our banding crew who have significant banding experience and bird extraction experience. And then we also have two banding interns who might have less experience and are learning really in a hands-on approach and increasing their net hours so that one day they can become lead banders. So you are out in the field and you just came across this wonderful mist net. It's beautifully set up, and it is totally filled with birds. So the first thing that our banders need to do when they pull birds from the mist nets and extract them is they need to identify them. So let's say that all of these birds were caught in your mist net. What an absolute incredible display. How many of them would you be able to identify on the spot? And I'll give you 1 point for every bird that you can ID. And I'll give you 20 seconds for this. Okay. This would be an absolutely astonishing net run if you got to pull all of these birds out from the same mist net. Um, but we've got going from A to K, a white-eyed vireo, a white-breasted nuthatch, a summer tanager, a northern water thrush, a black-throated green warbler, a blue jay, a sharp-shinned hawk, a hooded warbler, a golden-crowned kinglet, a black and white warbler, and a dark-eyed junco. Um, and most of these birds we can expect to see at least once in a typical season. There is one bird that really stands out here, and that's the summer tanager. So this is a bird that's known to breed much further south, and it's really unusual to get birds north of their breeding range, but it does happen, and particularly it happens in the spring when these birds are flying north to their breeding grounds and they catch a tailwind and they accidentally overshoot their breeding grounds. So when this does happen, we call these birds vagrants, and typically they disappear in a few days as they make their way back into their expected range, but sometimes they stick around, and that's really exciting. And occasionally we'll also get things like sharp-shinned hawks that are hunting other birds in the net. So it's really important that we go out on a consistent basis every 40 minutes to check the mist nets, because sometimes there are predators around that are interested in birds that are in the net. Okay, so you have gotten all of your birds out of the mist net and you've identified them, and now it is time to take them back to the banding lab. So the banding lab is where everything happens. It's filled with all the tools and the resources that our banders need to be successful and take all the data that they do. Many banding stations that are in operation today really started on a volunteer based passion project. So someone who was interested in researching and got it started. And when you're one of the oldest banding labs in the country, you definitely learn things as you go. So we've had some changes to our banding setup. One of them being that we got new floors, we got a sink, we even updated our computer system. But if you look at this picture from our throwback image and our banders from last season, and you can see the banding lab setup in the middle, the meat and bones of the operation has really stayed the same for the past 60 years. And so, as was done 50 years ago, what do you think is the very first thing that you want to do with a bird once it's back in the lab? You have 10 seconds. All right. The very first thing you need to do is ban that bird. So we do this first because if something happens, at least you'll have the ban number associated with the individual bird and any location information of where it got its banned. And even just that can be really helpful for migration purposes and understanding the movements of different species of birds and different populations. So each band that our birds get have a unique 9-digit number. And depending on the audience, you can think of it as a library card number or a social security number. And any and all data that's collected from other banding stations will be associated with that bird and that band for the rest of its life. And you can see here these strings up on the wall are the bands that we give our birds. And then down in the bottom right-hand corner are the bags that we bring the birds back from the mist nets to the banding lab in. And then up on the right is an image of specialized pliers that we use to put the bands on the birds so that we don't hurt their legs. And so each band is crimped onto the bird's leg to be the specific size for that bird. And what's really cool is that once a bird fledges from the nest, their leg is going to stay the same size for their whole life. So there's really no risk of putting a band on a bird and then having that bird grow out of that band, which is really cool. And then just as a little close up on the upper right-hand corner here, we've got a kinglet with its little legs dangling, and it uses the smallest band that we have, and you can see that it just loosely fits over the bird's leg. It's small enough that it's not going to fall over the bird's foot, but it's big enough that it doesn't restrict the bird's movement. And then on the left, the bottom left, we have a really good image of the bird in what's called bander's grip. So this is how we hold the bird so that we can take their measurements and put bands on them without hurting the bird. And it keeps their wings restricted and it keeps their legs in a safe position so that they won't hurt themselves. All right, so I think it would be fun for us to all see what it might actually feel like to handle a bird's leg to try to put a band on it. I'm going to give you 15 seconds. And if you are able to, I would love for you to go find a toothpick, a Q-tip, a pencil, a Sharpie, or, depending on where you are, either a cork or a glue stick. And I have a Sharpie here, so I'll give you 15 seconds to go grab one of those things, and you can start examining it. All right, so each of these items represents the diameter of a different bird's leg, and that's how we measure what size band we put on the birds. So starting with the largest band here, that would be if you were banding a cork or a glue stick, you would use a size 9, and our smallest, all the way to the left, is a toothpick, which is a size 0A. So if you have a cork in your hand or a glue stick, you are likely holding the leg of a bald eagle. The Sharpie represents the leg of a herring gull and uses about a 7A band. Our pencil is going to be a Cooper's hawk leg, so a little bit bigger than that sharp-shinned hawk that we saw a couple slides back. And then I'm not going to tell you what the Q-tip bird and the toothpick bird are, but I'm going to give you 10 seconds to write down your answers to test your bird ID skills. Okay, so our size 1 band, our Q-tip leg, is going to be something like a song sparrow. And then the toothpick represents a size 0A, which is the smallest band that we use in the banding lab. And that is going to be for something like a ruby-crowned kinglet. And typically at Manomet, we're only banding birds between a size 0A and a size 3. So something with legs between the size of a toothpick to a pencil. And we don't usually go much bigger than that. So thinking about that, there is one bird that we absolutely never band at Manomet. It is a native bird species, and we do catch it in the mist nets. What bird species do you think it is? You have 15 seconds. All right, if you guessed a ruby-throated hummingbird, you are correct. So this is the one bird that we would never band. In addition to having banding training, if you want to band hummingbirds, you need a lot of extra extensive training. So we're not trained or qualified to put bands on hummingbirds. And yeah, they just have teeny, teeny, tiny legs. And you actually have to shape the bands around their legs rather than the bands coming on rings like what we saw a couple slides back. And we do catch hummingbirds, so we'll weigh them. Sometimes we'll measure their bill length, and we'll also clip a teeny tiny piece of their tail feathers, just so that if we recapture a bird within the same banding season, we know that we've already seen that hummingbird. But other than a small tail clip, there's really no way for us to track hummingbirds from season to season. So, despite not banding hummingbirds, each year Manomet goes through thousands of bans on birds that use Manomet HQ as a stopover site, and I'm wondering if you know what organization is responsible for distributing the bans that are used at Manomet, but also at every single banding station across the United States and Canada. I'm going to give you 10 seconds. All right. If you guessed B, the U.S. Geological Survey, you are correct. And if you are also surprised, I was too when I first learned this. But it turns out the USGS is the parent branch of the Bird Banding Lab, which is where all of our banding data goes after we collect it at our banding lab. So the BBL has been operating since 1923 in partnership with the Bird Banding Office, which is its Canadian counterpart. And together, the two groups make up the North American Bird Banding Program. And so thinking about having over 100 years of bird banding data, I'm wondering how many million banding records are being managed since the NABBP's introduction in 1923. And I'll give you 10 seconds to write that down. Okay, if you guessed 77 million, you are correct. And this doesn't even include information on recaptures and resightings of those bands. These are just new bands that have been put on birds since 1970 or 1923. And each year, the NABBP gives out 1 million, over 1 million bands to banding stations across North America, which is pretty amazing. And I know that we all can't go start banding operations in our backyards, but you can see birds with bands. So pictured here is an eastern phoebe and a yellow warbler with these teeny tiny bands on them. And although we can't see them just with our binoculars, if you ever find a dead bird or a bird that's been stunned by a window and it has a band on it, you can go to reportband.gov and you can actually submit location data. You can submit anything you know about that bird and its band number, and you've now contributed a really important piece of data to this bird's life history. And you also get a certificate that tells you that you've done this, and it will tell you where that bird was originally banded. So that is actually so cool. And if you ever come across a band or a bird with a band, it's really fun to do that. So, once the band is put on the birds, there are a few other things that we like to do with the birds in the banding lab to connect or to collect important data on them. And so one of the stranger things that we do, if you're looking at the banding lab from the outset, is that we actually blow on the bird's belly. So I have a video of that here, and I'm wondering if you can tell me what can we learn from doing this? So what information can we get from blowing on the bird's belly? Can we tell how old the bird is, the sex of the bird, if the bird is ready to migrate, or the health of the bird? And I'll give you 10 seconds. All right, so one thing that we look for when blowing on bird stomachs are any signs of breeding, which can also help us to determine the sex of the bird. So something like a song sparrow that's pictured on the right-hand side here is what we call sexually monomorphic. So if we're just looking at it through a pair of binoculars, we can't tell if it's a male or a female. And before I jump into this, I'm actually going to give us all a quick bird anatomy lesson. So birds have one hole that they use for eliminating waste from their bodies and also for reproductive purposes. It's called the cloaca. And I couldn't find a seamless way to fit this into our trivia today, but I do think it's very important that everybody knows that in Rome, Cloacina was the goddess who watched over the Cloaca Maxima, which was Rome's main sewer system. So that's just something that I think we should all carry with us today. And so during breeding season, when we blow on a bird's stomach, we can look for two things that help us to tell if they're male or female. The first one is a cloacal protuberance, so that middle picture that we're seeing, the CP. And that is an enlarged cloaca that males will get during breeding season to more effectively pass sperm to the females. And then the other thing that we look for is the brood patch, which is the bottom picture. And that is something that typically appears when females are incubating eggs. So they will lose all of their belly feathers and their skin gets thick and blistery to better transfer heat to their eggs and nestlings. So if we see either of those are good tells that we're looking at either a male or a female during breeding season. The other things that we can look for are fat stores, and that's going to help us understand if a bird is ready to migrate and also the overall health of a bird. So, different from song sparrows, we have a common yellow throat over here, a female and a male, and they are sexually dimorphic. So, just from looking at them, we can tell that the top bird is the female and the bottom bird is the male. Um, and below, we also have a picture of a grumpy but very, very healthy common yellow throat. And this bird has fat stored in its furcular hollow, which is basically its collarbone. And because bird skin is so thin, we can see the fat stores underneath their skin. So that kind of yellow color that we're seeing right here, that's actually fat. And when a bird has a lot of stored fat like that, that tells us that they are pretty much ready to migrate, and it is also a good indicator of health. So everything about a bird's structure and their morphology is really designed to be efficient and light. And one of the few times throughout the year that they'll put on extra fat is when they're about to start migrating. And something like this common yellow throat might leave Manomet and fly nonstop to the Caribbean, which is really, really cool. Um, and then some other data that we take that I'm going to go through briefly, even though it's a lot of information, is we can look at a bird's skull. So just like a human baby, birds are born with soft skulls, and as they age, their skulls will harden up until they're about 3 months old. But during our fall banding season, any birds that we catch in the mist nets, we can look at their skulls to get a sense of when they hatch during the summer. And you can look at just how ossified those skulls are. So up at the top, we've got the least ossified. And then as the skull gets more ossified, it will create these little windows on the top of their head until you can't see them at all. I will say sculling birds is extremely challenging and it can change from species to species. But it's also really cool. And then the other thing that we look at are the plumage of birds and molt patterns. So if we're looking at the wings displayed on the right-hand side of the screen, I'm going to start with this yellow warbler where we can see three different types of feathers. So this is a bird that we caught in the spring, and we know this, and we can tell a lot about it from the different types of feathers that it has. So in blue, we have what are called the juvenile feathers, and those are going to be the first feathers that the bird gets after it fledges the nest. And then throughout the year, it's going to start replacing some of the other feathers. And so our banders are looking for what we call molt limits or distinctions between older feathers and newer feathers. So the next set of feathers that this bird got are its formative feathers, and those replace the juvenile feathers and are typically a little bit stronger and more durable. And then in red here, we have what are called our alternate feathers. So these are the newest feathers on this bird. And that tells us that because this is a yellow warbler, it gets a whole new breeding plumage. So it actually drops feathers to get more brightly colored feathers for the breeding season. So not all birds do an alternate plumage, but yellow warblers do. And then just some other cool things that are pictured here. Below, we have a white-throated sparrow, and you can see an example of pin feathers. So those are new feathers that grow in when, or new feathers grow in as pin feathers first, and then the sheath of the feather falls off and it expands into a full feather. And down at the bottom here, we can also see that this white-throated sparrow has these shorter tail feathers, and that tells us that something happened where the white-throated sparrow dropped some of its tail feathers, and then they grew in afterwards. So they're a different length than the majority of the other tail feathers. And then one quiz for you all is down in the bottom right-hand corner. I'm going to give you 10 seconds to guess which of these birds is an older bird and which of the birds is the younger bird. And these are eastern towhees. All right. So B is going to be the younger bird. And what we're looking for here is what we call a molt limit. So you can see on these coverts that it's a really dark black color, similar to the plumage of A. But once we hit this dotted line, we see this really light brown color, and those are going to be those juvenile feathers, right? So those are those younger feathers that are slightly duller. And eventually this bird is going to become entirely dark like A. What's really cool about plumage, though, is that this is something that you can look for when you're out in the field and when you're birding. And so in addition to telling maybe what type of bird you're looking at, the sex of the bird, you can also get a sense of how old it is if you can look for some of these color distinctions between its wing feathers, and also its tail feathers, which is really cool. A little extra info you can learn. So the last thing that we do when we have birds in the banding lab is that we weigh them. And before sharing how we do this, I do want you to think about how you would get a bird to stay on a scale to be weighed. And so I'm just going to give you like 10 seconds to ponder that. And this is an example. We use fairly generic kitchen scales. So this is the scale that we're trying to get them on. So just like all banding stations, there are things that have worked over time and we just stick with them. And the method that we use at Manomet is making bird ice cream cones. So each species of bird has a cone that is shaped specifically for them. And the birds are put into the cones head first. And this is a way to keep their wings from flapping and also to keep them calm. And then they get put upside down into an orange juice can. And I do believe that the can that we see here is orange juice concentrate, also from 1969. So some things really never change. But the birds are in these cones for no more than 10 seconds. It's a really, really quick process. And it's the most effective way to keep them calm and to get an accurate weight. And we want to get an accurate weight because that's a really important indicator of overall health, especially if we see large changes in weight over time. Okay, so if you have any of these things around you, I'd love for you to grab them. I grabbed 3 paper clips myself, just so that you can get a sense of what some of these birds weigh. And I am going to reveal, I want you to think about what kind of bird do you think you're holding in your hand? Because this is, whatever you picked, is the weight of a bird. So if you got 3 paper clips, you're holding a ruby-throated hummingbird. That is such a light bird. And then probably the heaviest bird in this array is going to be if you grabbed a Roku remote and 3 pennies. You're holding just about the weight of a blue jay. And then once we weigh the birds, which is the last thing that we'll do with them, we get to put them out this super cool window. And the entire banding process takes about 1 minute for each bird. So they're in and out of the lab really quickly. Everything that we do, we prioritize bird safety. So if for whatever reason there's something not safe happening, or the birds, like if the wind is moving the net lanes, we won't band. If it's too hot, if it's rainy, if there are environmental factors, we'll stop banding for the day. And what's really cool for us is obviously that we get to handle these birds and we get to see them up close, but because we're taking these long-term sets of ecological data, the impact of this work is a lot broader than just understanding one bird that comes through the lab. So we can start to look at long-term population changes, changes in timing of when birds show up, and start thinking about what other events and phenomena happen around us that impact bird populations. And beyond our local data, we're also contributing to global data sets that help us better understand the impacts of climate change on our bird populations, the impacts of um, buildings and habitat loss, and then also how we can support birds moving forward. So these are just some of the data sets that we get from our banding lab. Um, and rather than going through all of them, I'll come back to this at the end. And if people want to ask questions about the different population trends that we see, I'd be happy to talk more about it because this is really the information that we're going for when we take banding data. Um, but yeah, I'm going to skip my white-throated sparrow question, and if we have time at the end, I'll come back to it. Oh, but thank y'all so much.
Thank you, Claire. We do have some time for questions now, but I do want to ask the first question. What was the bird that you just skipped that you didn't show us?
This one?
Yes.
Okay, this is a white-throated sparrow. Um, and I mean, we could do the trivia question if we want. All right,
Because I know it's
so
exciting.
I'll give you 10 seconds. Which is the male? A or B?
Everyone has to answer, not just me.
All right. Well, I know you all are thinking about it. So what's really wild about white-throated sparrows, it's one of the coolest birds, I think, that comes through the lab. It's also the bird that we catch the second most numbers of. So gray catbirds are the um the birds that we catch the most, and then white-throated sparrows. And you actually can't tell which one is the male and which one is the female. So both male and female white-throated sparrows can either be what we call tan stripe on the left or white stripe on the right. And what's really cool is that white striped sparrows, either the males or the female, will be really aggressive and also will sing a lot more. And then tan stripes, so our birds on the left, whether it's the male or the female, will be better caregivers and they don't sing as much. And they always pair up with the opposite. So they keep that variation within the population. And it's just really cool because it's a bird that has two distinct characteristics and colorations, but either the male or the female can have that color combo and that personality, which is, I think, absolutely amazing.
Yeah, it is. So we had one question here in the chat that Evan kind of answered, but I still want to ask and see if you could give us an answer. And it's if there are concerns about capture myopathy in specific species, or maybe explain a little bit what that is. What's capture myopathy?
All right.
We're doing our best. We know Evan is in the chat. Evan is our director of land bird conservation who will deal
Yes.
with these questions. But yeah,
I will just kind of say what Evan said though. So most of the birds that we get are not prone to myopathy. And the banders that we have, like I said, we prioritize the health and safety of the birds. So our banders use methods that minimize handling. They have a really good sequence of what data they take and how long they collect that data, and the time that the bird spends in the banding lab is really minimal. And then also we prioritize smaller species, so we'll get them through the banding lab a lot faster, and hardier species we know can wait a little bit longer to be processed and then released back into the wild.
okay, I've got another question. I'm
Mhm.
wondering if you have any ideas on why the towers have crashed in all caps.
Yes, that is a great question. And we do have an idea. And so one thing about the data sets that we're looking at right now, these are very local and specific to Manomet. But if you are someone who lives on the Cape or you live in this region, if you were around between the 1970s and the 1980s, you will know that there was a lot of buildings that got put up and a lot of urbanization and a lot of apartment buildings, specifically here in Plymouth. And towhees are a bird that have a strong preference for pine barrens habitat. Um, and so what we think happened with the towhees is really just local habitat loss. So for the eastern towhees, we would have to now compare this data to what we're seeing nationally or with our Northeast populations to see a more general overall trend of these birds. But locally, we do know that this is because of habitat loss of our towhees.
Thanks, Claire. I have another one.
Yeah.
What can you tell us about catbirds and Manomet?
Oh man. I can tell you so much. Um, just catbirds in general.
Yeah, are they popular? What's going on with catbirds? Because everyone was commenting when you show your net and all the birds that could be there, there was no catbird. So there were some comments
This
about
is
that.
true. Yes. So catbirds are the birds that we catch the most here at Manomet. And that has a lot to do with a number of different factors. So one being is the location of our mist nets, right? So our mist nets go through shrub habitat, really leafy, greeny areas. And they're also lower to the ground. So we're not doing like canopy mist netting. We're not trying to catch anything that's above 10 feet in the air. And that is exactly where our catbirds travel to and from. They hop from shrub to shrub. So the mist nets are right in those locations. And then in addition to being a bird that might migrate through Manomet, catbirds also breed here. So we will get explosions of catbird populations in the fall because not only are we catching the adults that come through here during migration, the adults that are breeding here, but we are now also getting all of the fledglings that are just hopping around the brushes around the mist nets. Um, and we love them. If we could pick a mascot for at least the banding lab, I think we would pick a catbird. And if you don't know what a gray catbird is, they're pictured in the upper right-hand corner here, and they get their name because they make a little meowing sound in between, kind of trying to mimic other birds. They'll make a lot of gibberish sounds and then a meow and then a bunch of gibberish again.
They're amazing. One of my favorite birds, the Manomet. So we have some other questions here. There's a follow-up
Yeah.
about the trends. It says, aren't chickadees and blue jays seen more in urban and suburban areas? Why did they also crash? I assume the Vireo's loss was due to loss of habitat as well.
Yeah, these are really, really good questions. So I think I'll go through them one by one. So with the black-capped chickadees, they are a bird that they're non-migratory. So we see them here year round. Um, and what's kind of amazing about black-capped chickadees is that if they have the appropriate resources, they have they can lay up to 13 eggs at a time, which is unbelievable. And the way that they do this is every day they lay a new egg. And that first egg doesn't start incubating until the chickadee is sitting on its last egg. So that means that all 13 of those eggs are going to be hatching at exactly the same time. And so when we're looking at this chickadee population graph, those huge spikes are what we call eruption years, and those are years where the chickadees had ample access to resources and likely had huge clutches or laid more than 4 or 5 eggs at a time. Um, so if we take out our eruption years, chickadee populations have declined, but it's not quite as dramatic as it seems with those really big spikes. Um, so that's one reason why that seems really dramatic. What we can take from this graph, though, is that we're not seeing as many eruption years. So that might be an indicator that we don't have habitat with ample resources anymore, or that there's not enough food for chickadees to sustain larger nests. And then with our blue jays, kind of in similar vein, can I ask the audience something, Andrea? Or
Of
okay, does
course.
anyone know what a blue jay's favorite I think their favorite food is? Does anyone see blue jays carrying things throughout the year?
You are asking peanuts? I have
Oh,
three
peanuts. I love
peanuts. Yeah.
that. Okay.
Acorns.
Acorns. Yeah. So peanuts definitely, if we're thinking about food that we give them, and if we're thinking about food that they can find naturally, it's going to be acorns. So sometimes in the fall, you can see blue jays flying overhead with an acorn in their mouth. And if you see that, they likely have 3 or 4 whole acorns
Wow.
also in their gullet, which is unbelievable. Um, and because blue jays love acorns so much, it's thought that they follow the mast years of oak trees. And so a mast year is when an oak tree produces a large number of acorns, and blue jays will shift their populations based on those mast years. So also this could be an indicator that we're not having as many mast years locally, right? If we're not seeing as many blue jays. Um, but it also could be due to habitat loss, right? Like if we don't even have those oak trees, those blue jays aren't going to be attracted to this region. Um, I hope that answers the question of the blue jay. And then what was the other one? The red-eyed vireo. So,
Yes.
okay, red-eyed vireos are really fascinating. So the blue jays and black-capped chickadees, those are both, for the most part, we can think of them as non-migratory, right? We see them year round. Red-eyed vireos are migratory species, and they're also super long distance migrants. So, in addition to habitat loss, one thing that impacts these long distance migrants is the timing of when different things happen in different parts of the world. So, if we're thinking about a red-eyed vireo, um, and we're thinking about the climate here in, I'm going to use Massachusetts as an example because that's where I am, but we're thinking about the climate in Massachusetts, and then we're thinking about the climate in Central and South America, probably like Brazil, right, where our red-eyed vireos are in their non-breeding season. When red-eyed vireos are in their non-breeding range and things are happening up in Massachusetts during their breeding season, they aren't getting the memo that things are changing. So one thing that we're seeing here in Massachusetts and in the Northeast are these warmer winters. And with warmer winters, we see that our trees are leafing out earlier. And when our trees leaf out earlier, we see that our caterpillars come out sooner and start munching on those fresh leaves. And things like red-eyed vireos aren't necessarily getting that memo that they need to shift their timing a week earlier to catch that emergence of caterpillars that they really rely on, not only for themselves during migration, but when they're actually laying their eggs and they have nestlings, that's when they want to time that emergence of caterpillars. So if they miss that, then that really impacts how well they can care for their nestlings, and we're going to see their population start to decline. I will say that is one thought. There's probably many things that impact our vireo populations, um, but we call that ecological mismatch when things are starting to happen at times that they didn't used to happen at, and it's impacting the relationships between different species and how they rely on each other.
Well, the person that asked that question says that it's learning so much today, and that's why also the banding that we do, the maps banding that we're doing in the fall, it's so important. So we have two last questions. The last two questions before we wrap up this very amazing small set. It's what happens with birds that come in the net and they were already banded? Do you record the repeat bands?
Yes, that is an awesome question. And like, that is one of the most exciting things that can happen, right? Is if you get a bird that already has a band on it, yeah, we're going to record that information. And sometimes, in the case of the lovely catbird, we might get the same bird day after day in the mist net. Um, but what's kind of cool about that, so we'll still take that bird back each day. We'll record information on its weight. We'll record information on its plumage, on its molt. And what's kind of neat is that before migration, you might get a bird one day a gray catbird that's really underweight, and if you keep getting it back, you can actually watch it fatten up, which is really cool, and then it will disappear. So we do collect that data. If we get a bird more than once in a day, our banders know what band numbers they're using, so they'll just release them that day because there's no point in bringing the bird back to the lab to get the same data that we just collected an hour ago. And we don't want to stress the bird out more than we need to. Um, and then sometimes we'll get birds that are not banded from us, right? And that's super exciting. So we can get a bird that has a band from somewhere else in the world, and now we've just added this really important piece of data to that bird's life history and to the overall trends of that species, which is really, really cool. And I can't think of off the top of my head any that have come into the net lanes or into the nets recently from somewhere else, but I'm sure it will pop into my head at a random time today. But two birds I'm thinking of are a Baltimore Oriole and a Trails flycatcher that we banded here at Manomet, and then we got word that those got recaught in Mexico, which was unbelievable. So really cool to be able to see that they made it to Mexico and then also have it documented that we know this individual made that journey. And then one year we got a black-billed cuckoo that was recaptured. We banded it here at Manomet, and we got word that it was recaptured in Colombia, which was just so cool. So that's really kind of the most exciting part is knowing that your birds are going other places and we're getting that information.
Yeah, so there's also a comment or a question asking if we're doing MAPS banding in spring and the fall. And as far as I know, I think Evan is volunteering for MAPS every year. Is that happening this year too? You know?
Yes, and I'm just going to read, Evan put it in the chat, but in collaboration with Mass Wildlife, MAPS, which happens during the breeding season, banding at Miles Standish started in 2024, and we're planning to continue this into 2026.
And
From
finally, the last question, it's what volunteer opportunities does Manomet offer? I think that we can talk about the opportunities that we have for banding interns every year. What's
Yeah.
happening this year?
Um, yes, and so our banding interns aren't on a volunteer basis, but each season we get new banding interns, and this is a paid internship during those banding seasons. So from April 15th to June 15th, and then banders have the opportunity to come back again in August 15th through November 15th. And that is a paid opportunity where you can really apply for working in the banding lab. If you're coming in as an intern, you don't need a ton of handling experience. That's what this opportunity is for you to be able to get that hands-on training. And so you might just be interested in birds, you've been thinking about banding, and I encourage you to apply for that because that is absolutely an opportunity that you should take advantage of. And then sometimes we do have volunteers come through the banding lab and we coordinate that with our banders and also with Evan. So if you reach out to Kim Goggin at Manomet, she organizes all of the volunteers. She can connect you with whoever might be most helpful for coordinating volunteering.
Thank you, Claire. So yeah, we're running out of time. We could keep talking about this forever. We love birds. But I want to thank you in the name of everyone here for your time. We're getting great comments in the chat about how amazing this was. And a last reminder for everyone here, if you're new to Manomet, we're a nonprofit based in Massachusetts, but our work spans throughout the entire hemisphere, just like me, down in the south in Paraguay. So we're working from Alaska to Tierra del Fuego. And if you would like to stay connected, please follow us on social media, subscribe to our newsletter, and if you can, tell a friend, support us in any way you can so we can keep doing our science for many, many more years. So thank you, everyone, and see you the next time in March in our next small set. Bye.
Bye, everyone. Thank you.